Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show, bringing you another really awesome guest today, focused on some work that is really going to be creating a very unique and better tomorrow in many different domains. Today, we have the honor of being joined by Dr. Brett Kagan, who is the Chief Scientific Officer at a company called Cortical Labs, which is defined as a biological computing startup. And their focus is on putting live neurons into chips, ultimately to revolutionize computing as we know it, and ultimately help usher in what they refer to as the post-silicon age. And the potential applications for these unique tools of this so-called organic digital convergence are extremely broad, to say the least, including things like novel ex-vivo models for drug screening in various CNS indications like epilepsy and dementia, potentially a deeper understanding in general of just how brains ultimately organize themselves, obviously the inspiration for new machine learning methods, and ultimately down the road, potentially tapping the inherent intelligence of living neurons and their superior computing power, their low cost, to create ultimately real neuromorphic style computing one day. Dr. Kagan has his undergraduate degree in psychology from Bond University. It is Masters in Neuroscience at University of Queensland. His Ph.D. In cell therapy focused on the Neonatal Brain, University of Melbourne. And we are quite honored to have him with us today. Dr. Brett Kagan, thank you so much for taking the time to come on the show.
Well, thank you very, very much for having me. It's a pleasure to be here.
It's great having you. I really enjoyed, obviously reading about you and the company, but also just a lot of what you were involved in prior to Cortical Labs. And I would really like to start off just if you could talk for a few minutes about you, a little bit about your background, sort of where you grew up, how you got interested in just the neurosciences in general, but also a little bit about how, looking at some of your papers like that focused on the focal ischemic injury and the neonatal brain, how ultimately you moved from sort of that area of CNS pathologies to what you're doing now. I think that'd be a great way to start this whole story off.
Yeah, wow, that's a big question, not one that we normally, as a scientist, not normally one that's asked. I've got a bit of a strange background. I mean, for one part, you bring up sort of undergraduates. Like prior to that, I never even planned to go to university when I was younger. I'd always planned to drop out in grade 10 and join the military or do something down that track. And they talk about sort of brain development, especially in males, sort of the frontal cortex really coming online later. And I think I was a key example for that, because it was only when I hit about 16, 17, 18 that I really began to become interested in anything academic in the slightest. And I sort of started to look at a lot of literary theory. And that really kicked up my interest in how does the world work? How do we perceive and understand the world? And so ultimately I decided, let's give this school a go. Ended up taking to it quite well, getting a scholarship to undertake a double degree in law and psychology. So went to uni to do that simply because I thought it would be fun and it seemed a lot easier than a lot of the other alternative pathways. And again, didn't really know what I wanted to do. I was really just trying to explore. What I found out I really loved was, out of all of it, was the cognitive and the neuropsychology classes. And so I did a bit of a pivot. I did an honors in cognitive psychology, looking at memory. And then from that, I went, it's great that we have these theories about how the brain works in terms of the psychology aspect, what we see. And it's the idea of, like, right now, psychology, it's kind of like studying a movie, right? And I went, but how does a projector work? And so that's what led me to go do a master's in neuroscience and start to look at the actual cell biology and the cellular neuroscience aspect of it. And from there, I became really interested in, like, what are the tools that we have to be able to understand and use this better. And one of the big uptakes since 2008, when they sort of first developed the induced pluripotent stem cell technology and continued working on that, is this whole synthetic biology approach to understand it. And back then, although it wasn't terribly long ago, but things are progressing so quickly in the field, stem cell therapy was the main use that people were putting stem cell technology and synthetic biology for use. Since then, now, of course, and even back then, there was a lot of work just understanding how these cells arose and developmental work. Yep. But I found myself in a sort of more of a stem cell therapy lab. And so we're interested in that. But throughout all of this, I have always been really focused on the development of what leads to us as people, as humans, or even as like even for animals, what leads to their ability to exist and act and show intelligence in the world. And I remember having conversations, and I would sort of have a bit of a chip on my shoulder sometimes about the idea of artificial intelligence. And I would say, but look, how can you have true intelligence? And true intelligence has to be context dependent and adaptable. How can this arise when the basis of artificial intelligence, silicon, is inherently inflexible? And so when I was approached by Hon, and Andy, who are founding Cortical Labs, and they were saying, look, this is the idea we have. We need a neuroscientist. I was so excited to join, and that's kind of how I led here. It was quite a long and windy road, but it was always just this intention of how do we, what gives rise to us as people who can be intelligent beings in this crazy dynamic world.
That's excellent. Excellent. And Brett, on the website, you sort of take sort of novices like myself through a really interesting journey. You talk about as you introduce sort of us to this organic digital sort of convergence. You talk about the neuron, about how obviously we have 4 billion years of evolution here. It has all this amazing flexibility, self-programming capabilities. And you basically, hey, why don't we start with this as opposed, and you contrast, as you were just saying, artificial intelligence to more about what you're working on, sort of this theme of synthetic biologic intelligence. Obviously, you give lots of talks on this, but I was wondering if you could just walk us through, some of the novices that may be listening to this particular talk, a little bit about, in 2022, what the limits of silicon are, because you mentioned we need to surpass these. 2, just the introduction to what biologic computing means. And then if you could, thirdly, and this I'm asking for a lecture here, but just, again, introduce us to the theme of synthetic biologic intelligence compared to, and we've done a lot of shows on AI in the last year or so. It's been a very hot topic. Do a little compare and contrast there for us, just so we can all be in the same head state as you.
Absolutely. And look, I'm certainly not the right person to sort of give like an in-depth lecture or claim that I know the limits of let's say, silicon-based, machine learning-based approaches, or artificial intelligence, if you want to use that term. And frankly, I don't think we have truly reached what those limits are, but I think we are definitely starting to encounter at least the strong suggestion that there are limits. And I think this does contrast very strongly with a lot of the promises of what people sort of proposed in the past. Essentially, that if you just keep scaling up, the number of layers, and let's say like an artificial neural network, you will eventually get to human intelligence or consciousness or sentience or whatever term you want to use. And this will be a reoccurring thing you'll see, is that we actually don't have the terminology to even describe what we're trying to see in a lot of these cases. So we're going to have to make do with the best we can, and people are going to have to be understanding that, the context that we're talking about.
Sure.
But certainly we have seen it. So some of these limitations are that you can get amazing outcomes in some really special areas with, say, machine learning or artificial intelligence approaches. However, they do have some limitations, right? So they require a huge amount of data. Even with the best algorithms, even with the most sample efficient algorithms, they require a lot of data. They require a lot of sessions to run and to learn and trial and error for them to work. Opposed to, if we contrast as we go, humans, if I give you a binary choice, like pick the square or the triangle, and you pick the square and you get a little electric shock. You pick the triangle and you get, a chocolate. You're going to very quickly figure out what's going on here, right? It'll take you, 1, 2, 3 samples, and you've learned it.
Right.
Machine learning, not necessarily. They don't truly learn goal-orientated, and there are some, again, approaches looking at that, but they don't yet lead to the same efficiencies that you would otherwise see. So this is one example, the sample efficiency, and that's something that we've noticed and are exploring with our work. And seen quite some big differences in. Another thing is just power consumption. To run a machine learning algorithm, for example, huge power consumption, depending on the task you're doing. It can be literally millions of times more power consumption than what, say, a biological unit, what we run on. Our brains run off, some people dispute the number, but let's say approximately 20 watts. Versus, hundreds of thousands of watts potentially to train a machine learning algorithm to do an incredibly simple task, or more, again, depending what that is. And so this is like one really big factor. And so if you want to start claiming, well, we're going to introduce, AI or ML based learning in sort of a personal capacity for everyone to have their own custom intelligence unit, again, whatever the terminology you want is, how are we going to power that? We already living in a thermodynamically unsustainable environment as humans. We're aware of this. Climate change is obviously a thing that is happening, and we need to be able to develop better ways to adjust for that. Even if machine learning reached its claimed goal, this technology of the future that people claim is possible, is that going to be done in a way that's environmentally and thermodynamically sustainable? It's impossible at the moment, the current technology, to see how that could occur. And so if you're using a substrate like biology, which runs off, glorified sugar water, essentially, that could be a really viable way to introduce an intelligent system that is, sustainable and able to be introduced, in a personalized way for everyone. Now, again, of course, we're talking here distant future sort of, outcomes. But it's certainly feasible, right? That's the exciting thing about this. And I think that brings us to like the next point, which is this idea about generalized intelligence.
Yep.
Machine learning is quite fragile in the way that it's developed. It has stuff like catastrophic forgetfulness, where if you train it to learn a single task, you then alter that task in some minute way, the machine learning algorithm can break. And again, people are working on building robustness, but there's still a lot of work to be done. Biology, however, is incredibly innately robust. If you have a dog and you feed it in a blue bowl, and then you change and you feed it in a bowl that is, pink with butterflies on it, the dog still knows that's my food, I'm going to eat it. But of course, that's a very trivial example, because it gets more complicated than that. If you have a dog and you train it to be a guide dog for someone visually impaired, and you train it to avoid blue cars, it's going to know to avoid, the green cars, the red cars, whatever the cars are. Machine learning, not necessarily. So that's the other thing. Like, biology is by its nature generally intelligent, and we see this. This is a ground truth. We see it from everything from worms to flies to dogs to humans. We all show a level, and that level can change, but we show a level of general intelligence that we've not ever really seen in a machine learning and artificial intelligence approach, which leads the question is, can this be achieved with machine learning or artificial intelligence? Some people argue yes, and I'm not one to say like it definitely won't be, but we don't know yet. But biology, we do know it can be achieved. The only question is how, and how long will that take? And we don't have an answer for when this will be achieved, because there is a lot of engineering work to do. But we certainly know that it can be. So that's that's another really exciting difference. Yeah.
Excellent. No, I appreciate you laying that groundwork, just so that we know where we are and what we're dealing with. But spot on. I appreciate that, Brad. So then coming from that, and then obviously merging in a little bit of what you talked about in your bio and your early work with induced pluripotency and technologies of that nature. You're in essence starting with very basic cells, could be skin, blood, what have you, using the magic of Yamanaka and so forth to create sort of an endless supply of potential neurons. And with this capability, you have now moved forward in creating both really two systems that work together. One, DishBrain, where you, as you were saying, you have the magic solutions and the nutrients and so forth to create this interesting soft tissue. And then you're integrating it into what's called BIOS, Biologic Intelligence Operating System, where you work with the silicon together. Again, that was a very rudimentary explanation, but talk just a little bit about the background of this and what it's, because you haven't been around that long doing it, but it's an amazing amount of that you've achieved so far. Just talk a little bit about the history of this and in the creation of both DishBrain and biologic BIOS.
So there's a lot there to unpack, going all the way back to Yamanaka's and colleagues' work, and everyone else's work since it and before then.
Sure.
And so I think like one of the things to emphasize is like two factors that you bring up that I think I appreciate you saying we've done a lot, and we do work hard. We do work hard as a team, and I think there's two things. One, there has been amazing work in the hardware, the software, and what we like to affectionately call the wetware or the synthetic biology approach in the development over the last several decades. And of course, it's this groundwork that allows us to combine them and get an interesting result. And people have been trying and doing different aspects of this for the years as well. Even more specifically, they've been testing to see how do neurons respond to electrical stimulation. Since 2001, I think probably is the first most notable paper, but it could go back earlier than that. That people have been looking at how neurons respond with Hebbian plasticity, for example. Yeah. There's a huge amount of work that's been done, and all of that lays the groundwork to help us build up our results. And then, speaking of which, it's of course not just me, but we have a whole team that work with us. And, I do feel incredibly lucky coming to work because we have an amazing, diverse, multidisciplinary team that we get to work with, and this is like, I think one of the really key differences is we're not an academic lab, we are a startup. And, some people look at it and go, well, why would you be a startup? But the reality is that the way academic work is structured, I have never seen an academic lab, maybe they're out there, I've just never seen one, with this level of multidisciplinary integration towards a single shared goal. Academia tends to, by its nature, silo people. You have a neuroscience lab that will look at, let's say, synaptic plasticity. Normally, say, it gets even more specific, perhaps just between hippocampal neurons and subtypes of hippocampal neurons, and that's the thing they focus on. You get people who are brilliant in that. I'm not particularly brilliant at anything, I would say. Like I am, because of my history, like quite a generalist. And that's actually not ideal in academia, but in a startup world, it can be really useful because what it means is you can work and you can bring together ideas from a lot of different people and create something new and novel from that. And so, the term I love, I didn't invent it, I don't know who did, but is this idea of innovation through synthesis. And that's really how we have made our progress, is by bringing together all the parts that other people have developed, using our understandings to sort of pick the best pieces of that and accord it in, we have obviously put some thought behind it. And then seeing the output of that is sort of gestalt in some ways, right? The whole here is the greater than the sum of its parts. And that's, as I said, we have made some nice progress, and it's entirely the result of the chance to work with such a great interdisciplinary, multidisciplinary team and build off, considerable work that's been done in the past. In terms of how we're hoping to bring that to other people, you mentioned the BIOS, which we're calling as the Biological Intelligence Operating System, BIOS. So this is going to sit as a basically cloud-based platform, or it doesn't have to be cloud-based, it could be a unit, for people who want to use and test the system. And I think this is one of the other really nice differences between not being an academic lab, is that people talk currently, there's no questions about it, we have a reproducibility crisis in science. perhaps we always have, we're just more aware of it now. I suspect we always have, and we're just more aware of it now.
Right.
But and a lot of people are making brilliant work with that. But being a startup, like, our goal isn't to put out papers and see if people can reproduce them. Our goal is to make a thing for people to use. And so for us, we actually have to have a far higher level of reproducibility, because we need to say to them, it's like, hey, not only, never mind you trying to prove whether this idea is right or not. It's like, we want you to be able to do this, like, in your group. We're going to try and help you reproduce it, because our goal isn't to, hold some secret special knowledge, but actually to share that knowledge and to get as much community involvement as possible. And so, hey, maybe we've made a mistake or we've done things super suboptimally in our initial work. That's certainly possible. I think we've done, we've seen some nice results, but it's certainly possible that there's better ways to do it. And we are exploring some of those. But if you want to come on board and you want to use this system, like, we're trying to make it as easy and accessible for you as possible. And so essentially, this is in some ways like trying to return back to the start of your question, where I sort of said the results are because we've had amazing people to work with and amazing results from other people to build off. We now want to go back very quickly and sort of say, hey, and we want to see other people make amazing results building off what we've done. And that's sort of the idea about why we've developed, or developing this BIOS system for other people to be able to access.
Yeah, it's a very elegant approach, and the concept that, as you were mentioning about the reproducibility and getting it out there to, obviously you guys are, I take my hat of it. You're extremely smart, but yes, I mean, doing it in this collaborative way with all the other people at the forefront of this domain is, it's refreshing, and it's nice to see that. But just as a slight sort of side segue for a moment, because as I mentioned, as I was sort of looking through your previous work in the peer-reviewed literature, there was this really interesting paper that it popped out to me. You wrote it in 2021, and it was entitled Integrating Levels of Bone Growth Control from Stem Cells to Body Proportions. And, this just got me because, one of the most fascinating areas to me is sort of, as a generalist myself, that as I've looked at this whole area in terms of regeneration and repair and so forth, is this whole concept of form, right? And for size control, that, these are not things, it's sort of one of those magical areas. These are not things that either one gene or multiple genes really say, put a brain in here and shape it a certain way and keep it a certain size and all that. There's a lot of sort of biophysics and everything behind all that. And thinking just about the brain in general, when we're embryos and it starts developing it whenever, 6 weeks out, and then becomes this 100 billion or so neurons in a certain size and structure and all that. Can you say just a few things about sort of what you're learning? I know it's early and you're only dealing with sort of tens of thousands of neurons now, but any interesting insights regarding form and sort of structure as you are working with DishBrain and DishBrain 1.0, 2.0, and so forth. Any interesting insights there that sort of connect to some of your stem cell work in terms of form and proportion?
Yeah, well, that's a big question as well. So yeah, basically that work was done during a postdoc that I did after my PhD with Alberto. Alberto is one of the most industrious and hardworking scientists I have ever come across, and it was great to work with him. Although I did find myself missing the brain, and it's funny comparing, let's say, like bone to brain in terms of research, because they are actually like very different ends of the spectrum in many ways. Because a lot of work has been done establishing and understanding a lot, let's say, of the gene pathways of the regulation. There's still a lot of questions to ask about, let's say, bone growth regulation. And there's some really interesting things still there to be unpacked. But there's so much work that's been done compared to the brain, right? Especially like, let's talk about form and the complexity of form compared to, let's say, bone versus the brain. There is certainly still complexity. There's still a lot of complexity with bone, but they're not, I'm trying to think of the right way to word this, because they are adaptive and dynamic in response to the environment, but to a far less extent, I might get an email with people disagreeing with me from this, but I would say to a far less extent than to the brain, especially when you consider how neurons can change their states and their role in a given state within a, compared to, let's say, chondrocytes or anything else, much more dynamically and diversely, I would say, than other cell types. And there's still so little known in scope of how much we imagine there is to know, how much we understand that there is to know when it comes to the brain. So the questions we're asking are quite different in many ways, and in many ways they're the same, which it's this continuum, of course. So in regard to answer your question like a little more specifically, when we look at, let's say, form in the brain, it is inherently tied to function in some ways, but it's hard to exactly say what that is. And it kind of comes down to this idea that we're currently investigating, which is to say, what is a computational unit in a brain? And so if you look at the bone, right, like let's say a growing bone, growth plate, whatever the case is, and you can say like, there's not really a computational unit, but we can say like, what is the functional unit, let's say, of that? That's really discrete and it's quite obvious. And you can be like, look, the cells are doing something and then they're working as a whole, and that's what's going on there, right? It's pretty nice and neat. I feel that you could say that there. But with the brain, what exactly is a unit in the brain, a functional unit or a computational unit in the brain? And like, I think it's fair to say, like, we don't really know, right? People started off, and you go back, like, you can go back like to the start of it, and people would be like, oh yeah, this part of the brain does that. Memory is located here. Look, Descartes, saying, oh, the pineal gland's the seat of the soul. Like, you can go back quite far and it gets, kind of more amusing from our perspective now, but they'd sort of said, look, there is a localization of function in this whole area. And to a degree, we now know like that is true. You can map certain areas, the motor cortex, you can map it quite well and it does relate. But what we've also found is that if you damage an area, you can get a rewiring and other areas will fill in. You can get this active recruitment. You can have a plasticity that continues to surprise us. The more we investigate plasticity, the more we find that brains are able to be plastic. And then you go deeper and you say, well, something like intelligence or memory, which we've always known is fairly distributed, even if there are, for example, like, hippocampus is important for the formation of long-term memory, but we know there's a lot more than that going on. Is it a single neuron? Is it two neurons? It has to be a network of neurons. And we just don't know. In our work, we're starting to investigate it. And, we kind of came with this hypothesis of, it's probably a single neuron could be a computational unit. And what we realized is, well, that doesn't seem quite right based on, there's some more work coming out on this. But, they will be able to show fairly soon. But we've gone, it's not quite right that it's a single neuron is a functional unit, but it's also not quite right that a single neuron isn't a functional unit. Okay. What it seems to be is that, yes, a single neuron can do some interesting stuff, and so can two neurons working together, and so can three neurons that then have this, exponential growth that's happening in terms of the network formation. And then a group of 10 neurons also work between each individual neuron and each individual collection of, subset of that. And so it's this massively exponential growth of complexity that I believe, at least, at this stage, is going on in the brain. And it makes it really hard to sort of wrap a head around and be like, this is the link between the form of a neural system, what it looks like structurally, and the actual function of a neural system, how it functions. And like the most simple example of that is anyone who does work with neurons in an MEA can sit down, they can have a layer of neurons, and they can have two identical layers. And you will look at them and, okay, sure, they'll look slightly different because neurons are arranged randomly. But then more or less you'll look at them and you go, yep, those are both biological neurons on a dish. And you put them into an MEA system and you measure them for electrical activity, and it can be chalk and cheese. There is pretty much, within bounds, if there's cells are dead or dying, of course they have no activity. But as soon as you have two healthy cells or groups of cells on a plate and you want to check their electrical activity, there's pretty much no link that we've seen, or very little link that we've seen to sort of predict based on structure alone or form alone, whether or not they have function. Okay. I don't have an answer for that. I know some people are investigating it and some people coming up with some answers, but it's a mystery. And, I could dive down into all the hypotheses that, people are investigating about that, but it's a fascinating subject, and it's one that I hope that in time the neuroscience will catch up to other areas of science that have had significantly more time to investigate that and actually be able to provide an answer. And this comes up with something I was talking with Andy, who's one of the founders and co-author of the paper, about just the other day. He's bringing up this idea of what was he calling it? Constructive content.
Okay.
Which is to say, he was sort of defined it. I thought it was a brilliant, beautiful concept. When we're looking at trying to define a system, you can give it a word. Let's say in this case, the word sentience, which we controversially used in this paper to describe what we saw as a system.
Sure.
And what we were trying to do was use that word with the idea of constructive content, right? Which is to say that can we give it a trait to identify how one might reconstruct this trait? In this case, it had to meet a set of criteria, and that if it did, then it would therefore be useful in such a way. And I think that's where we need to really be heading and focusing in the neurosciences, is how do we recapture and reproducibly recapitulate the link we're seeing between the physical nature of what we're seeing and the functional outcomes of the system. Hopefully that wasn't too long an answer, but it's a great question. It's a great question and a very difficult one.
No, it was spot on, and I appreciate you going that in depth with it. No, I appreciate that, Brendan. Okay, so a couple weeks ago, in vitro neurons learn and exhibit sentience when embodied in a simulated game world comes out. Press all over the world about this one. And in essence, again, you have DishBrain, BIOS, and via certain electrophysiological simulations and so forth, you, in a matter of a couple of minutes, basically, BIOS learns Pong, a version of the 1970s arcade game Pong. Talk, obviously everyone can, and we'll put a link to this paper in the bio of the show as well, but talk just a little bit about what this experiment was all about, what the teaching process was in terms of, as you're saying, learning this particular goal in such a short period of time. Walk us through a little bit of this, if you would.
Yeah, so I'll just make a quick correction, if I may. So
sure.
The BIOS is the system that we're currently building.
Okay.
We didn't actually have a BIOS system on top of the current sort of DishBrain system.
Sorry.
They're kind of different. BIOS will replace DishBrain because DishBrain was essentially, it's a prototype. It's a proof of concept. It was put together with minimal resources, and as I said, amazing team who could work under these conditions. But that makes it slightly inflexible, a little janky. It's early work. So BIOS will come on top, basically replace that and be a far more user-friendly, flexible, adaptive, and hopefully reproducible. In that we'll be able to see the same or similar learning effects across a number of different games quite easily. Currently, it's quite difficult to program new games with the DishBrain system. So that's really exciting. It's going to sort of change the game and how this work's done. In regards to how we were running the DishBrain system, essentially, it was a great question, right? Because we sat down and basically when Hon and Andy brought the problem to me, they were interested in engaging in the neurosciences. They'd looked at some work by, there's a researcher called Isamura over in Riken Institute, and there's another researcher who's, I think, retired now, Steve Potter, who's also done some work previously in this area, and they've kind of looked at these papers and have gone, all righty, look, there's some interesting suggestion that neurons can engage in, semi-complicated behavior such as blind source separation, which is also known as like the cocktail party effect, and that they can show quite high degrees of synaptic plasticity if we close the loop. But how do we actually tap into and get a goal-directed or, what we call intelligent behavior out of these neurons? And it's a big question because there is no dedicated reward system, right? It's not like people, we often use the word initially, we've stopped using that word usage because it gets confused, but we often said reward or punish, and what we really mean was like incentivize, decentivize the system. Because you can't really reward or punish this, right? They don't have a dopamine system. And well, they might have a dopamine system, but if they do, we don't know how to access it individually. We can't shoot in a drug, with the time scale we need. quick enough to reward them for doing the right goal, and then give them something negative. I don't know what you would do to punish them exactly. Some sort of toxin or something to be like bad neurons. Yeah, like I don't know what you would do that. That's the thing. It's like, even does dopamine really reward a neuron? Like, in our brains, yes, but not in. So it's such like a difficult question. Like, how do you reward a group of neurons? They're not conscious. They don't have a feeling of like, I should behave in this way. They have no incentive to play Pong unless we make an incentive for them to play Pong. So we had to figure out, like, at this really deep layer of like information entropy, why does a neuron want to do what it does? And luckily, as I said before, amazing work has been done previously. So we looked at, and for example, in this work by Isomura and blind source separation, he found that this actually accords with the free energy principle, which was developed by one of our co-authors, Professor Carl Friston. Now, the issue a lot of people had with the free energy principle is that say, well, it's a great idea, but it's very hard to test. Because essentially in a whole human, if you want to test this in a whole human, we have a lot of compensatory mechanisms. If you want to test it in animals, again, there's a lot of stuff going on. So this was kind of a limitation. But if you can break it down and test it in just cells that don't have all these compensatory mechanisms, that are just responding to the most rudimentary and coarse stimulation you can provide them, information, as it were, that you can provide them, it is possible to actually isolate the various factors that may be ongoing to try and get some suggestion. And so that's essentially what we set out to do. We sort of went, well, look, this is a great theory. It gives us some useful implications to test. It has a lot of theoretical sort of backing to it. And if we can find evidence for it, it would be actually very useful, because it would provide the sort of fundamental basis for all intelligence. And we could use that as a push lever to sort of encourage neurons to behave in a way that we want, in a fairly, and I say fairly, like relative to what people have done previously, complex manner. And so that's what we set out to do. Now, I'll be perfectly honest. Carl and the people, he works with, and some of the people we work with, like Adil and Anjali, they've done amazing work developing the free energy principle. I kind of went into it quite naively. And I went, well, it's great, but I also was reading the other, the concerns people had with it. And I went, I don't know if the free energy principle, I think there's a lot of there to it. I can agree with a lot of the justifications. But intuitively, to me, I was like, I don't know if this, like, just to me, I was like, I don't know if this is really going to be the thing that explains it. I don't know if this will really get learning. But what we were prepared to do is we went, look, rather than just guessing on the theory, because we can go back and forward on that forever without an answer, let's just empirically test it. Let's come up with a test that we can all agree, yes, this may work, yes, this may not work. And whatever the results are will tell us, as long as we've done it in a way that everyone agrees beforehand, this is a good way to test it, the results will be super interesting either way. And, I was, it seems, based on the paper, quite wrong. The free energy principle actually did promote learning in a fairly, time, restrained manner. We saw a difference between the first 5 minutes and the last 15 minutes in a game pretty rapidly. And that was super exciting to see. And, it's not necessarily surprising to see, because again, the arguments for why the free energy principle could be a viable source for, sort of neural activity, self-organization, however you want to call it, they are strong arguments. And I think that it'll be really interesting to see where we can grow from here, using a more empirical focus for systems neuroscience and computational neuroscience development than what people have, as a general rule, been engaging in. And I think that's fascinating. And I don't think it's, necessarily because people engaged in that more theoretical work haven't been eager for empirical research into this to sort of test these, although there certainly are some, that would rather just be able to make claims that never are tested. But I think the majority by far love the idea of being able to test these theories. But it's just that the technology hasn't been there to do it in a tightly controlled empirical manner.
Right.
And so when you can start to break down those different barriers, you can actually start to unpick what it is that makes, let's say in this case, 800,000 neurons on a dish respond in one way versus another.
Brett, alongside this paper, you also wrote a another paper that was entitled "Neurons Embodied in a Virtual World: Evidence for Organoid Ethics," and you brought up this issue early on about sort of the troubles in defining consciousness, sentience, and so forth. And I think, some of these organoid models going back, this has always been something that's sort of discussed on the side. Then, well, do these models ever start thinking, and and where does this eventually head? Obviously, the early days, and you need this. You think about these things, but maybe they're not the core focus of everything that goes on there. But why did you write this paper? And I mean, are people asking you in these early days of the work that be very concerned about this? Because I Christoph Koch on the show a few months ago, talking about sort of supercomputers and will they ever become conscious, and all the reasons not. But here we're dealing with something a little different, and I just like to get your couple minutes on again why you wrote this paper at this time, and any sort of external feedback you're getting per the whole sentience component of this.
Yeah, and can I just say, your talk with Christoph, he's such a legend. The work he's done is amazing. It was I enjoyed watching your show.
Thank you.
With him. Yeah, absolutely. Look, there's a few reasons why we've chosen to take that pathway, and we're continuing to develop with a number of independent bioethicists this work and taking this approach out. One of them is simply because it's important. Too often people have gone to develop technologies without considering the use cases, the approach of the development, and it generally doesn't end well. We're trying to be different. We're trying to be collaborative. We're trying to be constructive. We're trying to make sure that the technology is developed and eventually applied responsibly. We don't want to go, outpace, and to the extent that people get scared of this work. There's no need to be scared of this work. There's nothing scary about it. It offers huge ethical gains, but if you don't understand the work, people's response when they don't understand a thing is to be afraid of it.
Yeah.
And we don't want that to happen because there's no need for it. And so in this case, this paper was a response to another paper by a group who was sort of arguing that there are cases already for organoid research where it should be just considered impermissible to discuss that work. And we kind of had to start and say, well, hold on. How can one say that this is impermissible before one even understands what the work is?
Right.
And, I must say, we've actually, chatted with some of those authors since, and, there is actually a real drive to work together to actually figure out how we move forward. And I think, if more researchers, in the sort of basic science approach were engaging in this, I think we'd have a lot more collaborative approaches. And I think what happens is that when biological or basic science, let's just say researchers, whatever science you're in, you want to progress your field, at the exclusion of all other opinions, people's reaction is, oh, we need to put a brake on this because we don't understand it. But if you engage more with the community, you can actually come together and you can figure out where are the best use cases, what are the advantages. So I'll give you an example with our use case. There was concerns with the organoid work that these things may somehow, I have no idea how, be conscious. Again, there's no evidence whatsoever for this. But okay, that's your concern. And so people say, well, they may be conscious, therefore let's adopt a precautionary, they call it a precautionary principle, which means if there's a chance it could be conscious, we should just stop. Mm-hmm. Now, we kind of went, that's not the best approach, because what if there are ethical gains that far outweigh the very, very remote and unlikely risks? For example, classic example, a lot of the work in identifying approaches for the Zika virus were discovered through organoids.
Yeah.
Huge ethical gains. If you can use organoids or our system, for that example, as a stand-in or replacement to reduce or to replace animal testing, that's a huge ethical gain. If you're worried about consciousness in an organoid, which again, I must stress, no evidence whatsoever that this is the case. How can you not be worried about consciousness in a mouse or a monkey?
Sure.
I mean, these are things that certainly feel, respond, can care, maybe not how we do, but they're living animals, and yes, animal research is necessary, but if we can reduce it, that is a huge ethical gain. And so we kind of wanted to bring up and say, look, this is an interesting area of research, but we can't respond to it with sort of slippery slope arguments of fear. We need to respond to it by looking at all of the approaches. And that's why we're continuing to do that now. And I think that's really exciting, because the more people you bring along, the more use cases you can come up with this, and good ethical use cases for it as well. And that's why we take this approach, and we'll continue to take it. And welcome, I'll say for anyone watching, we welcome anyone's involvement. We can't obviously work with every single person as much as we try to, just time. But if anyone is interested in reaching out, or you have a concern, or you have whatever, like, we will do our best to sort of work with you and step through this to make sure that, things are, lead to that utopia, not the dystopia.
Yeah.
Right. That's the goal.
No, yeah, that's spot on. Yeah, no, it's refreshing. So, but again, speaking about the use cases, when I introduce things, they're broad. So, you can do CNS drug discovery, you can create neuromorphic computers, in-depth study on sort of brain organization across different time spans and so forth. And now I'll come back to you. Obviously, I have no neuroscience background, so this is going to be more kindergarten style question here. But other potential things when you talk about use cases that you could potentially put into this system, and I'm just wondering, things, because neurons aren't all, as you work with bone, and so they're not the only thing that has complex electrochemical communication in our bodies. And I was thinking, we've had folks like Dr. Mike Levin over here at Tufts on the show a few times, talking about all the membrane potentials and potential sort of central nervous system information processing that goes on across these trillions of cells. Any sort of other applications out there in terms of not just not a DishBrain, but other organoid systems that you may want to study, potential, I don't know if you want to call them intelligences or not, but and then for that matter, other species that may not have a central nervous system like we do. I know Mike used to put slime molds on similar types of devices and see how they agglomerated together and studied, how they communicated and all that. I know this is a sort of the weird question, but any of the weird stuff that has come up along the way that might be neat one day to play around with in the bios.
Yeah, well, Mike is absolutely another sort of legend in the space, and we love following his work. And yeah, also brilliant the work him and his group are doing. So look, we are unnamed Cortical Labs. We are focusing on cortical and brain, neuroscience-based, approaches. That's not to sort of downplay the incredibly interesting stuff coming out of these other areas, but again, sort of bring right back to what I was saying at the start. We have ground truth for highly complicated dynamic activity, or behaviors, intelligence, whatever you want to call it, that comes from the brain. Could some level of these stem from another of our other biological substrates, or whatever the case of pseudo-biological substrates? In case of slime mold, I mean, it's biological, but also it's a bit different looking at that. I know there's some interesting work going on there. Yeah, of course there could be, but is it likely to reach the complexity that we see from a functioning brain? Right. I don't think so. I'd love to be proved wrong, because that would be super fascinating. But we've never seen it, right? There's no slime molds moving around in anything that approaches real time, solving mazes or whatever, like it's not happening. Right. And so, that's to say, like, for our focus, we're going to continue to focus on the neuroscience. And there's still so much that we don't understand about how the brain works and how neural systems come together. I don't think we're going to run out of that anytime soon.
Not that you should. Yeah. And what do you have to change the name of the company anyway? Because it's a cool name. Yeah. Brett, anything else hot coming up for 2022 as we get into 2023? Obviously, we're watching for more publications, but conferences you're going to be presenting at, places where potentially we could listen to you, meet you. Anything else that's happening that you might want to mention, either with you or the company? Please take the floor.
Oh, thank you very much. Well, look, the goal going forward now is to basically expand, refine, and extend what we're going to be doing. So we're going to be aiming to scale up how we're doing things.
Mhm.
We're going to be able to wanting to refine and better understand how do we make these systems work. So one of the things we commented in our paper, of course, is that essentially like the hardware, wetware, and software, although some of the reviewers were very kind, and they said it's the most advanced system that's been built for this. And I think technically that might be true, but that doesn't change the fact that it's still a very basic system. It's still a prototype. And so we really compare it to like early nascent computer industry, like the first transistor, right? It was, if you've ever haven't seen a picture of the first transistor, go Google it. It'll probably shock you. It's quite large. It's janky looking. Now, many thousands of them sit inside a cell phone. It's amazing the progress we've made in 70 years, and that's the progress we're looking forward to. And sort of in the paper, we said like any improvements on any of these axes is likely to yield improvements. And so we're aiming to improve across all of them. And we have really nicely established pathways forward. On top of that, as I mentioned before, we're taking this highly collaborative approach. We're going to be trying to make our technology as accessible to as many people as possible. One, because it's good science, and two, because it's going to be part of our business model, much in the same way as Intel or Nvidia sell chips to people to work on how to make better chips.
Yep.
It's the same approach that we're taking. That's sort of the model that we're following as a platform technology. We're going to be reaching out. I'll be talking at a neuromorphic conference. Yeah, so what we're trying to do is work with a bunch and extend out to a bunch of different sort of industries and research areas. So, for example, I'll be presenting in the NanoG conference on neuromorphic materials, devices, and circuits and systems.
Whoa, that's a name.
Which is going by I think the NewMat CAS conference, which should be in, I think it's in November. Sorry, not November. I think it'll be actually December or January, early next year. So that'll be great. We're working on, reaching out to work with obviously different types of neuroscientists. We're working to look with more machine learning researchers, and we want to sort of extend across all of these. And I think the really exciting thing is, I'm glad it's great. And we honestly didn't expect a reaction from people that we got for our first work. It's great that people care. And, we're excited that they're excited. But I think the really exciting stuff is what happens next.
Yep.
When people pick up. And actually, you can see that this is happening across the world now. So, there's the organoid intelligence group out of Johns Hopkins, led by Thomas Hartung, Lena Smirnova, and a number of other very talented researchers. I think they're going to be looking at similar to what we're doing, but in a more focused case with organoids specifically.
Mm-hmm.
There's the Mind in Vitro group out of the University of Illinois, led out of the University of Illinois, which is also in the States, which is going to again be looking at similar topics. There's the Neurochip Consortium over in the EU. All of these research groups that are, and I'm sure there's others as well that we're not even aware of. All of these research groups that are popping up in a very collaborative way. What discoveries are we going to make? It really does feel like a new frontier and a new research field that's popping up, and I'm really excited to see what these groups start producing. We're hoping to be able to work with any of the ones that want to work with us, and to see what can be built out of it all.
It's going to be an exciting future in this domain and everything you're doing. I mean, it's just really fascinating work, Brett, and I just I will continue to follow you and your company and really as you move these technologies forward, and rooting you on as you do it. Again, for everybody that is going to be listening to this episode of our show across the various podcast networks or watching on our YouTube channel, again, you've been listening to Dr. Brett Kagan, Chief Scientific Officer, Cortical Labs, doing really amazing things to move forward the organic digital convergence and help usher in this post-silicon age. Brett, it was I want to take the time to thank you for taking the time to talk to us about all these issues and all the work you're doing. Obviously, thank you for doing this work. And as we like to say on our show, thanks for helping to usher in this better tomorrow for all of us via the work you're doing. It's really very impressive, and I will continue to follow it.
Oh, again, thank you so much for having me, and hope you found it interesting. And yeah, likewise, thank you just for the chance to communicate it.